Hey everyone! In the last video, you learned about the different parts of a chromosome and how a karyogram can be produced using the differences between different chromosome types. In this video, you'll learn about the different systems that we use to describe the location of a gene within a genome. And this time we have just one question. And our question is, how is the location of a gene described? A given region of DNA on a chromosome is called a locus. You can see the sequence of that region here. You can also think of a locus as a place on a chromosome. Multiple places on a chromosome are called loci. A locus can be described in two ways. First, it can be described by its location on its chromosome when that chromosome is fully condensed as a metaphase chromosome. Locations of all the loci of a chromosome described by where they are in their metaphase chromosome make up a cytogenetic map of that chromosome. Second, a locus can be described by the number of base pairs between that locus and the beginning of the chromosome. Each base pair in a chromosome is numbered, which provides a coordinate of each locus. For example, the TGM3 gene starts about 2.3 million base pairs from the beginning of chromosome 20 and ends about 2.34 million base pairs from the beginning of that chromosome. These coordinates mark the physical location of this gene. The relative order of all genes on the DNA molecule of the chromosome and their base pair coordinates relative to the beginning of the chromosome make up the physical map of that chromosome. This might be hard to imagine, so later on in the video we'll take a look at an actual physical map of a chromosome in the human genome. Let's look at how to describe a locus in a cytogenetic map. Here is a simplified cytogenetic map of chromosome 3. Each chromosome has two arms, each on either side of the centromere. The shorter arm is always called the P arm for petite or small, and the Q arm is always the larger one. The chromosome is further divided into bands, which correspond to bands visible when the chromosome is stained and viewed under a microscope. Band 1 is always the band closest to the centromere, and you count outwards from the centromere. Each band is further divided into subbands, and each subband is divided into sub-subbands, visible only with high-resolution microscopes. If we wanted to describe the cytogenetic location of the ULK4 gene, which is found here, we would say its location is 3p2.1 for chromosome 3 on the P arm in band 2, in subband 2 of band 2, in sub-subband 1 of subband 2. As practice, write down the chromosome arm, band, subband, and sub-subband of the following locus. The locus is 11q14.1. I'll give you a moment to pause the video and give this a try. Okay, so the cytogenetic location is chromosome 11 on the Q arm in band 1, subband 4, in sub-subband 1. And if you made the attempt, well done. The physical map of a chromosome is made up of the nucleotide sequence of that chromosome and the location of each locus within that sequence. The physical location of a locus is described as the number of nucleotides it is from one end of the chromosome it's on. This is a more precise way of describing where a locus is. As an example, let's take a look at the Human Genome Browser, which shows both the physical and the cytogenetic locations of loci. Okay, so what you're looking at here is the UCSC Genome Browser, and we're taking a look at chromosome 13. And we see here is a gene, it's called MED4 or MED4, and in this browser, one of the tracks is the coordinates of each location on the genome. And we can see that the MED4 gene starts about here and goes to about this point. And so it starts at about 48,070,000 base pairs or so from the beginning of the chromosome and goes to almost 48,100,000 base pairs from the beginning of the chromosome. So it's a pretty small gene. This genome browser also shows you the nearby genes, which is also part of the physical map of a chromosome. We see the NUDT15 just upstream of MED4, and if we scroll over a little bit, maybe we can see another gene. Or we can zoom out a little bit, continue scrolling, and here's another gene, the ITM2B gene. So physical maps also show relative proximity of genes and gene order in addition to their base pair coordinates. Now, nice thing about the Human Genome Browser is it also shows us the cytogenetic location within a cytogenetic map, and that's shown up here. Here we can see the centromere of this chromosome, and we can see we're on chromosome 13, and it shows us in parentheses the location on the cytological map of the location that we're looking at in the genome browser. In this case, it's on the Q arm of chromosome 13, band 1, subband 4, and subband 2. We can also see the location on the chromosome over here by this red line. We can see it's on the Q arm, and if you weren't seeing this red line here, it would say band 1, subband 4, subband 2. And you can think of the coordinates of a gene sort of like the address of that gene, the address of a house within a city. And in this metaphor, the gene would be the house, the coordinates would be its address, and the chromosome is the city. So hopefully that makes a little bit more clear for you what a physical map of a chromosome is, best exemplified by the UCSC Genome Browser.
Okay, so by this point, you should be able to describe how the location of a gene within a genome is described using the two systems we discussed. And as always, if you can't, feel free to watch part or all of the video again. Now in the next video, we'll look at how chromosomes change as cell division proceeds. See you then.